Before we begin the presentation, I would like to introduce today's speaker, Rik de Greef. A full speaker bio can be found in the blue widget on the bottom right of your console. Rik is the Executive Director at Certara Strategic Consulting. He currently leads the group based in Oz in the Netherlands. He was trained as a PK/PD scientist at Leiden University. His key interests are in integrating model-based components with overall clinical pharmacology and clinical development deliverables throughout the development trajectory. Rik, welcome to today's event. I'll now turn it over to Rik to begin today's presentation.
Okay, thanks Marcella for this introduction, and welcome to everyone. Good morning, good afternoon, good evening. I'm very happy to be able to present this webinar to you, which has as a topic immunotherapy in oncology and how modeling and simulation can be applied in that area. And of course, this area is of keen interest to a very broad community these days because of the promising results obtained in terms of immunotherapy in oncology recently. Here's an overview of my presentation. I will start with a brief introduction into the topic of immunotherapy in cancer, and quickly focusing on a specific target in that area, PD1, and I'll explain a bit further what's that about. Also, we'll be discussing current development pipelines targeting this specific target. And then I will go on to discuss modeling and simulation, specifically in oncology. How can modeling and simulation be applied in that area? Then moving on more specifically into immuno-oncology, clinical development there is slightly different from your typical clinical development. So there are some key challenges there, but it also brings in opportunities for the application of modeling and simulation in this area. And then I'll close actually, the majority of the presentation will be a few examples of the application of modeling and simulation in immuno-oncology. Okay, introducing immunotherapy in cancer. The concept of the cancer immunity cycle isn't necessarily completely new. It's about the interaction between the endogenous immune system and cancer, tumor cells, which can be visualized as a cyclic process in which antigens on tumor cells activate the immune system, which then will attack the tumor cells, leading to further death of tumor cells, and this can be considered as a cyclic process. In that process, there are not just stimulatory aspects, but there are also mechanisms that try to regulate this whole process so that it doesn't go out of hand. However, in case of a developing cancer, this immune system apparently isn't doing a very good job. And immunotherapy has been looked into and investigated as a means to stimulate and restore the immune system to fight cancer through this cancer immunity cycle. This concept isn't new. Immunotherapy has been in development for several decades already, and three main types can be distinguished. One is cellular, in which case immune cells from a patient would be removed, and immune cells specific to the antigens on the tumor are activated, cultured, and ultimately returned to the patient with the aim to stimulate the immune system. Another one is cytokine-based, whereby essentially endogenous cytokines are supplemented with treatment with exogenously administered cytokines. And the one that I'll be discussing today is antibody-based immunotherapy, where we have proteins that are essentially antibodies against certain antigens, and through that can stimulate the immune response. And I'll discuss this in more detail on the next slides. Antibody-based treatment in cancer isn't necessarily new. So I have here several examples. Rituximab is probably the first one. Trastuzumab is a very well-known Herceptin targeting HER2. The first one really targeting this cancer immunity cycle was ipilimumab, which targets the CTLA-4. and was approved in 2011 for the treatment of melanoma. And this really works on that cancer immunity cycle. It turns off the inhibitory system that interrupts T-cell activity, so in that sense it reactivates the immune system. And it can be regarded as the first immune checkpoint inhibitor. It shows very clear activity, but also a considerable level of immune-related side effects because it targets the immune system in a fairly systemic way. I'll get back to that. Because in this cancer immunity cycle there are multiple targets, many of which have a general role in the immune system, with then the risk of systemic immune-related adverse reactions. However, there are several targets that are more specific to the interaction between T cells and tumors. And one of those is PD1 or PDL1, PDL1 being the ligand of PD1. So PD1 can be considered a key immune checkpoint in the development of treatments against cancer. What does it do? It essentially downregulates the immune system by preventing the activation of T cells. So it's in a way a regulatory system preventing the immune system going out of hand. So it reduces autoimmunity and promotes self-tolerance. And this inhibitory effect of PD1, which is activated through the interaction with PDL1 or PDL2, its ligands which are expressed, for example, in tumor cells. The inhibitory effect is accomplished by, on the one hand, promoting the apoptosis in antigen-specific T cells, but simultaneously also reducing the apoptosis in regulatory T cells, so the T cells that regulate the immune system. And in that fashion, PD1 and the interaction between PDL1 and PD1 inactivates the immune system. Well, there is an opportunity now to use this system to reactivate or stimulate the immune system, and that is by blocking that interaction between the PD1 receptor and the PDL1 or PDL2 ligands. And that can be done in two ways, essentially. On the one hand, antibodies that block the PD1 receptor, so PD1 targeting antibodies. On the other hand, there are also antibodies that bind PDL1, the ligand that's expressed on the tumor cells. And both types of therapies are being investigated these days. Because in the meantime, there's a fairly substantial clinical pipeline for PD1 and PDL1 antibodies. The most advanced are two PD1 antibodies, which both have been approved in late 2014, one from BMS, nivolumab, and the other one from Merck, pembrolizumab. But there are several others in late stages of development, among which two PD-L1 antibodies and one additional PD-1 antibody. And as you can see, there are several also in more early development stages. So quite a number of companies have shown a keen interest in this area, also are collaborating between smaller biotech and bigger pharma in order to try and find optimal treatments targeting this important checkpoint. And it's not just these compounds, because let's say a dozen of compounds in clinical development seems quite overseeable, but actually what's making this an even bigger effort is the large number of combinations that are investigated. And this is as an example of the program that Merck is conducting, and I've taken this from a Merck corporate presentation where they describe investigating more than 80 combinations with their PD-1 antibody, pembrolizumab. And those combinations range from combinations with standard therapies, so cytotoxics, targeted therapies, so those are compounds that target specific proteins in the cancer growth cycle. Immunomodulators, other types of immunomodulators beyond PD-1 antibodies, as well as vaccines. So you can imagine trying to look at 80 different combinations is quite a challenge, and it can be envisaged that modeling and simulation can play a role in trying to optimize combination treatments and leverage data across combinations. But there are definitely good reasons why companies are putting all their efforts in these potential treatments, because actually the efficacy data for PD-1 antibodies look very promising. And here are two examples from the melanoma indication, where both nivolumab on the left side, and what I'm showing here are overall survival curves, so it shows overall survival over time. So on the left side is nivolumab, on the right side is pembrolizumab. Both show clear improvements versus standard of care, where nivolumab was tested versus a pre-immunotherapy standard of care, and pembrolizumab was compared versus ipilimumab, so the first immune checkpoint inhibitor. Both showing clear improvements in terms of overall survival. What's also interesting to see is that the level of PDL1 expression on the tumors is an important factor in the response to treatment. So patients that are so-called PDL1 positive consistently, and they're shown here in this graphic in the red bars, consistently show better outcomes than the PDL1 negative patients, which are shown here in green. And this also implies that the best responses for these types of treatments can be expected in those types of cancer with a high expression of PDL1. One of those is melanoma, non-small cell lung cancer is another one, and you can definitely see that targeting those cancers that have the highest expression of PDL1 gives also the best results. Okay, that's for the introduction into immunotherapy and specifically the PD-1 target. Now on to modeling and simulation, how that can be applied in oncology. And I'll start with a more general description of what types of questions can be addressed through the application of modeling and simulation, and specifically also PK/PD types of modeling and simulation. Of course, oncology is a special area, but the types of modeling that can be applied are not always specific to oncology. They can also be envisaged to be applied in other types of indications. So first of here, the prediction and characterization of human PK. Semi-physiological or more physiological approaches can be used to predict PK at the site of action or in other populations, going from animal to man, but also between different types of patients, both adult and pediatric. The characterization of biomarker responses in early clinical studies can help to define doses that you would expect to be biologically active and help determine a dose range for further clinical testing and efficacy studies. The translation of preclinical data, and I will show an example of that in terms of efficacy. In oncology, that would then often be in terms of looking at mouse xenograft data, again to support clinical dose regimen setting. The characterization of variation in drug exposure, this is important ultimately from a labeling point of view, characterizing the impact of so-called intrinsic and extrinsic factors on drug exposure. And typically in oncology, often these types of effects are not studied in dedicated clin pharm studies, as may happen in other indications, which means that population PK analyses need to be applied based on sparse sample data from patient studies. Furthermore, and this is probably the key one in oncology, is the characterization of tumor size responses of the treatment. Tumor size in itself is somewhat of a surrogate marker. It's often not the primary endpoint that's looked at because that's survival. But looking at tumor size responses can really help establish the optimal dose regimen and a therapeutic window of your treatment, and also can be used as an early marker for survival. For example, as a tool in the stratification of patients, looking at the early tumor size response, understanding whether a patient is likely to respond or not. Furthermore, of course, on the other side there is the safety profile. So establishing exposure-response relationships for safety will also help optimize your dose regimen from that perspective and help, together with the efficacy data, help establish a therapeutic window. And altogether, these models can be used in the optimization of trial designs as we are used to in other indications, looking at different elements of trial designs. And then last, also in oncology, there's the opportunity to apply model-based meta-analyses of publicly available clinical trial data to get an understanding of the competitive landscape. What are the opportunities for your own compound to differentiate itself from compounds already in the market or under development in the same indication? If we then try to visualize a framework, and I've borrowed this from the Uppsala University group, a framework of modeling and simulation in oncology, then you can visualize it like this, with a very central place for the tumor response model, which is fed by a PK model through exposure metrics. Those same exposure metrics also feed a biomarker response model, on the other hand, an adverse effect model. And altogether, they help us establish a survival model and predict either progression-free survival or overall survival over time. And such an integrated modeling and simulation framework can of course be gradually developed and will have a variety of applications over time, and will have different focus areas over time during development of a compound. An even more broader view is presented here, where again there's a central tumor size model, but that then links to an even broader gamut of model types, be it preclinical, even in vitro data, clinical biomarker data, clinical tumor growth data, survival data. They all can be tied together through a central tumor size model, and really in this fashion utilize all data in a holistic manner, trying to optimize and support key decisions during drug development. I must say that this holistic view, it's very interesting. I haven't seen it in practice in this truly holistic fashion, but elements of it definitely, there are examples of probably every one of them. Okay, then moving on to the challenges and opportunities that are brought by the specifics, I would say, of clinical development in immuno-oncology. And to get those specifics clear, I first would like to present you a picture of the standard clinical development trajectory that everyone probably is very familiar with for a standard indication, non-oncology. This is typically how a development trajectory could look like. Starting off with single ascending dose and multiple ascending dose study in phase 1, then moving on to patient studies, probably a proof of concept study followed by a dose finding study in phase 2, and then the set of pivotal efficacy and safety studies in phase 3, which lead up to the submission of an NDA and hopefully ultimately the approval of the NDA. And in parallel, a range of healthy volunteer studies is conducted to address specific clin pharm questions. Well, this is a very phased and sequential approach with clear cut points, milestones, if you will, and those milestones are logical places to inform the next phase through modeling and simulation, and have these so-called learn confirm cycles, whereby we learn from the modeling of the data obtained in the initial studies and confirm them further on with our next study. However, if we then look at clinical development in immuno-oncology, and this is an abstraction of one of the recent programs in that area, which wasn't much more than essentially 2 studies, both big studies, but one being a phase 1b study in patients, starting off with the dose escalation followed by dose expansion, looking at several doses still. And as you can see, the pivotal efficacy study started already during the conduct of that initial study, and actually even the submission of the NDA, or actually a BLA, took place midway during the first study, and while the pivotal efficacy study had hardly started. This means that the timelines are very condensed, and the classical phases that we are familiar with are not discernible here. And as a result, there's no clear separation in different learn-confirm cycles, which essentially means that as a pharmacometrician, you should be on board with the team on a constant basis and continuously be there to apply modeling and simulation to address a variety of questions. Also, no healthy volunteer study, so the characterization of clin pharm characteristics really should be done as part of these efficacy and safety studies. So to sum up some of the key challenges in immuno-oncology clinical development from a modeling and simulation, but also general practical perspective. As I said, the timelines are very condensed. And there's a good reason for that, because it's really driven by the promising efficacy of these compounds, which resulted then in the opportunity for fast-track approval with regulatory authorities. Also, lack of early clinical biomarkers. There are no good biomarkers at this point to predict efficacy of these treatments. So there's a limited opportunity to inform those regimen selection with early clinical data. Also, and of course, there's a tremendous opportunity there for these treatments to be applied in a wide range of cancers, but it also means that the treatments have to be tested in all these cancers, and which from a modeling and simulation perspective presents a challenge, but also an opportunity to try to leverage data between indications. Can we learn from one indication to optimize our dose setting in another indication? And as already alluded to, endless opportunities in terms of potential combination treatments. So also there, need to think about how to effectively use available data to optimize combination treatment dose setting. So one could see the application of modeling and simulation in immuno-oncology to build on essentially, I would say, 5 pillars. And I've described those 5 here on the left side of the slide. On the one hand, it's clinical translation, so from preclinical to clinical, the early dose setting where modeling and simulation can play an important role. Then it's the dose justification later on, or confirmation at the point of submission of your application to the authorities. The general clinical pharmacology characterization for submission purposes. The support of combination treatment development. And last but not least, also, this is a competitive landscape that evolves at a very rapid pace. So trying to keep track with that and in a quantitative fashion, have a handle on what's out there in terms of data on the compounds being investigated in this area is I think of key interest. In this specific presentation, I'll show examples for 3 of these pillars. So the first 3. One more translational, the other one, I would say, empirical exposure response types of analyses, and last but not least, a POP-PK analysis to support the clin pharm characterization. Okay, moving into those examples and going to the first there. This is a translational example where modeling and simulation was applied to support the early clinical dose setting for pembrolizumab, which is the Merck anti-PD-1 antibody. And as background, the phase 1 study was already ongoing. In that study, classical dose escalation was performed. In that dose escalation, no MTD was observed. As part of the dose escalation, a biomarker assay was used, looking at the ex vivo IL-2 stimulation to assess PD-1 receptor modulation. So it's a means to look at target engagement. And you see a plot here, which presents the results of that biomarker, where the AL2 stimulation ratio in a decreasing fashion shows an increase, is associated with an increased target engagement. And you can see as concentrations increase, indeed there's a signal. As you can also tell, data are quite variable. Also, there was no understanding yet of what the exact connection would be between target engagement and efficacy. So overall, this was considered insufficiently robust to base any dose setting on. So complementary method was sought after, and translational modeling of mouse xenograft data was applied in a somewhat sophisticated way, which I'll try to explain here. So a translational model framework was set up, and you can see here it was built on both data-driven elements, which are depicted here in yellow, as well as literature elements. So essentially existing models taken from literature, which are depicted here in red. So we had data in mice, xenograft data, including PK data in mice. We had some PK data from this first study in men. We used a literature PBPK, physiologically based PK model to characterize the exposure of the tumor to the antibody. Then we had some data on receptor occupancy of the antibody and in the tumor. And we noted that upon treatment, some upregulation occurred. So we had a model developed on the mouse data for that specific aspect. Then tumor growth was based on a literature model again, and that ultimately was used in a fitting fashion initially to fit the mice data. So we had an integrated model that linked all these components, and we fitted PK data, receptor occupancy data, as well as tumor growth data with this model framework. And I'll show you the literature pieces that we used. So this is the PBPK model, a model developed by Shah et al. To describe the distribution of antibodies, not necessarily just in tumors, but across the whole body. So it's really a whole body PBPK model. The great thing about this model is it has been developed for a range of species, including mouse and humans. So that then allows for translation between species of exposure. In our case, we have simplified the model to focus specifically on the distribution to tumor tissue and to enable a translation of the data that we had in mice to a predicted tumor tissue exposure in men. The other literature-based element was the tumor growth model, and there we used the Simeone model, which by now is really an established model to describe tumor growth in xenograft models. And the translation to the human situation there was done by replacing the growth rate in this model with estimates of human melanoma tumor growth rates that we obtained from literature. And also, since the Simeone model and xenograft data in general is represented as a volumetric tumor size estimate, whereas clinical data is based on longitudinal measures, so looking at the diameter. So we made that conversion along the way when we did our translation. So this is then how our translation looked like, and working towards a simulated human tumor response. So we essentially switched from the mouse PK model to our human PK model. We had our literature-based tumor exposure model, in which we actually also replaced the target binding constants, which we had from in vitro data, both in mice and humans. So we incorporated the human constants. We had the upregulation piece, and then there was a tumor growth piece in which we replaced the growth rate with typical melanoma growth rate from literature. And that was then used to simulate human tumor responses under a range of dose regimens with pembrolizumab. And here we have summarized those simulation results. So in this particular simulation, we predicted the tumor size effect upon repeated treatment after 6 months. And we categorized the tumor size responses in a manner that's similar to the classification done by the so-called RECIST classification, where tumor size effects, changes from baseline, are categorized in several categories. And here we haven't exactly used the RECIST categories, but categories that are similar to those. In our simulations, we included multiple scenarios because we weren't completely sure about the best way to scale our tumor growth and the drug effect from mice to men. So for that, we established a range of scenarios. And the simulations that I'm showing here are essentially integrated across all those scenarios. And what the simulation showed, and you can see here, red would imply progressive disease, so essentially no treatment effect, stable disease, whereby the tumor size remains approximately stable, an intermediate effect, a partial effect, and a major effect, which is close to what's otherwise called a partial response or complete response. As we can see is that the dose range from, let's say, 2 milligrams per kilogram onwards would produce a maximal effect. There's no further improvement to be expected beyond those doses. So based on this work, 2 milligram per kilogram was decided to be taken forward as the lowest dose to be tested clinically. The MTD hadn't been established, as I said, and the phase 1 study had gone up to 10 milligrams per kilogram. So that was the dose range, 2 to 10, that was brought forward in the efficacy studies. So translational work here was really used to pinpoint that lower end of the dose range to be tested clinically. Okay, moving on to the next example, which actually ties it into this one to some extent. Looking at dose justification through modeling and simulation. And I'm first looking at the efficacy side of things, and then later on also briefly at safety. Well, typically the approval of oncology drugs is based on survival data, and then, as you can see in, for example, summary basis of approval for oncology drugs, exposure response analyses of survival data are used in support of dose justification. However, the recent immuno-oncology treatments that have been approved received their initial approval already on tumor size-based response data, or overall response rate, ORR. And logistic regression of that overall response was performed versus exposure to justify the approved dose. And I'll show the examples here. For both nivolumab and pembrolizumab, this is taken from the review materials of the FDA. And logistic regression was performed of the objective response data. And for both compounds in the dose range tested, so the exposure range explored, no significant exposure response was detected based on the analysis of ORR versus exposure. You can see different exposure measures were used for both entities, probably driven by the initial choice made by the sponsor. However, this ORR provides, in a way, a somewhat limited view on the effects on tumor size, because it looks at a single point in time and really at the categorical response. So it's a yes or no for each patient, and that's what you have. So modeling of the complete tumor size response over time for all individual patients should provide a better insight in the impact of exposure and other factors on the response. However, if we then look into immunotherapy, the response patterns there are somewhat different from those seen with classical chemotherapy. In chemotherapy, you often see tolerance developing, so a regrowth of the tumor. We haven't seen those types of patterns, at least not to a large extent in immunotherapy thus far. So alternative approaches have been explored to characterize tumor size dynamics in immuno-oncology. And one is shown here, again, for pembrolizumab, where the starting point for model refinement was a model developed by Laurent Claret, one of my colleagues at Certara, and this is, in the meantime, a quite established model to describe clinical tumor size responses, which describes the change in tumor size over time as a function of a tumor growth rate and a tumor kill rate. And the tumor kill rate is influenced not just by the size of the tumor, but also by a tumor resistance development component, which develops gradually over time, as well as drug exposure. So drug increases essentially the killing of the tumor. Well, in the pembrolizumab data, we did note some specific features that led us to considering changes to this model framework. Just to point out that the tumor size measure used here was the sum of longest dimensions, or SLD, of the target lesions, which is a quite common measure to use. But some remarkable features were, of course, the overall decreasing trend, which is good, but with a wide variety in patterns. Limited regrowth, hardly any regrowth, I would say, with some patients plateauing at certain minimal tumor size, but not necessarily showing any regrowth. Also, the data set, and this is not uncommon to oncology data sets, showed some early dropouts, so patients that already dropped out directly after the baseline, potentially because of a progressive disease, but no data recorded, so it cannot really be confirmed. But also, on the positive side, complete responses, so patients that achieved such a good response that no detectable tumor was left. But that's also a missing data feature that we had to address in our model. So the refinements to the model that we considered to accommodate this variety of patterns were the following. Initially, we looked at fixing the tumor growth rate because we did not see this regrowth occurring. We wondered if we could actually estimate a growth rate. Ultimately, we didn't need to fix it and we could estimate it. We did remove the tumor resistance rate because of the lack of regrowth observed. And in order to address these patterns that plateau at a certain minimal tumor size, we also introduced what we call the deep tumor compartment. And I'll show you in the next slide how that actually worked. As for the missing data below the detection limit, so the complete responses, we replaced the first response below the limit of detection with a value of half the limit of detection, and subsequent observations below the limit of detection we deleted from the data set. So that's a very simple, if not simplistic, imputation method, but it worked well. We did some sensitivity analysis and did a good job. The most important element that we actually introduced as a refinement was the definition of different subpopulations with distinct tumor size response patterns. I'll show those here. So what we used was a so-called mixture model in which we defined 3 types of submodels, all based on the same generic model developed by Laurent Claret. And altogether, we then defined 4 mixture groups because one of the submodels we also had actually 2 types of responses. So just going through them, the first submodel was the so-called escape model, in which only tumor growth occurred, so no tumor kill. So those were patients that just showed an increase. Then there was the so-called monophasic, which is essentially the Claret model without the tumor resistance component. And we distinguished there fast and slow patients, so either with a faster tumor kill or a more slow tumor kill. And then we had this biphasic submodel where we defined a deep tumor compartment, which essentially is untouched by growth, kill, drug effect. It is there, it stays there. and it's thought to represent essentially that piece of the tumor that's either not accessible to the antibody, will remain there. And if we then looked at how our model behaved compared to the observations, then we indeed saw that this mixture model could characterize the diversity in growth patterns. Of course, important to note that our predictions here on the right side reflect what we call population predictions. They don't take, it's individual predictions, but they don't take into account residual variability. So the more erratic patterns that we see in the observed data are not completely reflected in the predicted data. They show more smooth patterns. Of note, the early dropouts that we had in our data set were addressed by the so-called escape model. So those patients that just had a baseline measurement and dropped out directly thereafter, we assigned to this escape mixture group. We essentially took the conservative assumption that they were fast progressors. And when we then utilize this model to predict tumor size effects and to look at the exposure response, so essentially, and that translating that to dose response, we did not see across the dose range tested from 2 to 10 milligrams a huge effect of exposure on the outcome. As you can see, there is maybe a slight hint to less progressive disease with increasing dose, but it all falls within the margins of error. And this essentially confirmed the appropriateness of the lowest dose tested in the program, 2 milligrams per kilogram, as the therapeutic dose for pembrolizumab. I should recognize that this is the work we presented at PAGE in 2014. In the meantime, additional data has been accrued, and while the developed model characterized the data well, it had some complexities through the use of this mixture model, which made the covariate search as well as simulations a bit more cumbersome than desired. So with the expansion of the dataset, alternative approaches have been explored, and I would like to refer you to a presentation by Anna Kondic et al. At last year's PAGE, where she demonstrated a simplified model which actually had similar performance characteristics, but still was able to characterize that variety of tumor size response patterns. Okay, so that was the efficacy side of things. Now briefly on safety. So the first immune checkpoint inhibitor, ipilimumab, was associated with immune-related adverse events, as I mentioned, probably because it has an effect in a more systemic fashion on the immune system. However, logically, these types of AEs, next to general AEs, were then of interest in the review by the FDA of the 2 anti-PD-1 antibodies. And one way to look at it is to look at the relationship between the occurrence of these adverse events and exposure. And different methods actually were used. One more descriptive, and again, logistic regression was also used. And I'm summarizing those here on this slide. Wherefore, nivolumab, comparisons were made in terms of exposure, so it's C average, between patients with grade 1, 2, so lower grade general AEs, and those with grade 3 or higher general AEs. No clear difference in terms of exposure seen there. Similarly, for the AEs of interest, so the more immune-related AEs, exposures were similar between the patients with or without such an AE. For pembrolizumab, similar types of AEs were looked into, but then through a logistic regression, so linking the occurrence of these AEs versus, in this case, AUC as exposure measure. And here you see the general AEs, and on the right side is the AEs of interest. Across the board, for both antibodies, In the dose range tested, there was no indication of a relationship between exposure and the occurrence of AEs. So which essentially means that the therapeutic window for these compounds is fairly wide. Okay, moving on to the final example, and briefly discussing the key role for population PK in the characterization of clinical pharmacology for these immuno-oncology compounds. As I already mentioned, no dedicated clin pharm studies have been performed in these programs, and PK was assessed mainly through sparse sampling in the patient studies. So population PK was used really also to provide a general PK characterization, looking at aspects as dose linearity and time dependence, to assess the impact of intrinsic and extrinsic factors, and of course also to provide individual exposure estimates for all the exposure response evaluations that I already have shown you. So the population PK for these two approved PD-1 antibodies, not completely surprisingly, is really very similar. Both are well described by two-compartment linear models. For both antibodies, body weight is a key covariate on both clearance and volume of distribution. Then, as a result of covariate searches, a variety of covariates came up as being statistically significant because both analyses were supported by large data sets. However, none of these covariates were found to have any clinical significant impact. But what's most important to see here is actually the impact that population PK has had in terms of labeling for these compounds. The PK, as well as the drug interactions sections and special population sections of these two antibodies, are heavily supported, if not solely supported, by population PK, which is quite exceptional to see, although we see it more and more these days, and I think it's a good direction, because the population PK really allows you to have an integrated look at all PK data accrued during development. All right, that was my final example, and brings me to the summary of my presentation. I think I've shown you there is an opportunity for impact for modeling and simulation in the area of immuno-oncology. I've shown the examples of the early dose setting, where through our translational work we have been able to openly inform, especially the lower end of dose selection. Finally, at the end, towards the end of development, which was just 1 year later, justification of the dose, really providing an integrated foundation, looking at efficacy and safety for the therapeutic dose. The support of the clin pharm sections in the label through population PK. And then there are definitely opportunities there in support of combination treatment development, where I think a more systems pharmacology approach is probably required to really understand potential synergies between combination treatments. And last but not least, this is a very quickly evolving competitive landscape, so model-based meta-analyses should help define differentiating factors between the different treatments that are out there in development. So with that, I would like to thank you for your attention. I'll now happy to move on to any questions that you may have. I'm looking to my colleague Huub, who's moderating that.
Thank you, Rik, for a great presentation. My name is Huub Klein. I will moderate the Q&A session. As you have seen, there's a Q&A box on your screen, which you can use to submit any questions to Rik. So let me start off with the first question. That was a question about the tumor growth data. We were wondering which method was used to handle the data below LOQ in the tumor growth modeling.
This is the clinical
tumor growth modeling.
Yeah, so as I mentioned, essentially a simple imputation method was used, whereby from the time onwards that a patient decreased below the limit of detection. The first observation that happened, the value was replaced with a value half the limit of detection. So say the limit of detection was 5 millimeters, it was set at 2.5, and then the remainder of the observations were essentially deleted from the data set as considered, let's say, not providing any additional information beyond that first point. So what then happens is that the predicted curve will be driven below the limit of quantification already by that first imputed point, and then the points thereafter, your model will predict a value below the limit of detection. So I hope that answers the question.
Okay. Next question from the audience is about model-based meta-analysis. Are you aware of any examples of the application of model-based meta-analysis to bridge across indications in immuno-oncology?
No, at this point, I'm not aware of definitely not of a published application of the published application of model-based meta-analysis to bridge between indications in immuno-oncology. In a way, of course, our translational analysis actually did bridge because it looked not just at predictions in melanoma, which I have shown here, but in parallel also did the same for non-small cell lung cancer. So there was a bridge, let's say, through the preclinical arena. But definitely, I think there's a tremendous opportunity here. Of course, data are still, it's still early days, and there's still limited data across the variety of indications that immuno-oncology treatments are being tested in. But as data accrue, there will be a tremendous opportunity to learn and see if we can connect between indications and through establishing an integrated framework help, for example, dose setting or patient selection in other indications.
Okay, thank you for that. Another question from the audience is about co-treatments as covariates in your efficacy models. Have you tested for co-treatments in those models?
No, so in the efficacy model, we did not include factor for co-treatments. We did in the POP-PK just for one specific type of treatment, the combination with glucocorticoids. So it's an immunosuppressant often used in combination. We did not do a formal test in the efficacy model. I know that in the review by the FDA, actually some exploration in that direction was done. No effect was seen though.
Okay. One more question from the audience. You mentioned about PD1 and PDL1 expression. That it can play an important role. Have you considered incorporating it in your model?
Yes. In practice, actually, in fact, in the melanoma dataset, the initial dataset that I've shown the analysis for, we did not yet include it. Also in melanoma, PDL1 expression seems to have somewhat less of an effect. In later analyses, non-small cell lung cancer, it has been included and actually also showed a clear effect.
Good. One final question then, and that's about the Xenograft animal tumor model. These models generally are immunosuppressed or immunocompromised. Are these still valid models to test agents that are targeted on the immune system?
Well, it's actually a very good question. I think our work has shown that there is a predictive value because what we've seen is indeed that even though we did not make any translational adjustments to address the fact that the immune system would be different between mice and men in this case, we were able to predict the dose range associated with efficacy, and that was ultimately proven right by the clinical data. I'm not sufficiently a biologist to go any deeper, unfortunately, into responding to this question.
Okay.
Any further questions? I think we have, maybe one.
Maybe one last question. Let me see. Is the FDA requiring any specific modeling in the review of immuno-oncology products?
No, I wouldn't say specific. There are specific requirements. Of course, I am not the agency, but from what I've seen in the summary basis of approval, they're looking at immuno-oncology products similarly as to other oncology products. Definitely, I think there's still, even though these compounds are very promising, show good efficacy, there are still the same requirements in terms of the characterization of clinical pharmacology. So the question-based review that's applied in other programs is equally applied in immuno-oncology. So as a sponsor, you will need to supply a good characterization of the clinical pharmacology of exposure-response relationships. And I think both nivolumab and pembrolizumab have shown that that is feasible even with such a condensed program.